So hopefully I can project enough for you all to hear me. Yep. I'm Kristen Lindquist. As a representative of both the Monhegan Museum, where I work, and Monhegan Associates, for which I'm a board member, I want to welcome everybody to our second in-person event in our Summer Art and Ecology event series. This is a collaboration between the Monhegan Museum and Monhegan Associates. And before we start, I want to thank the Monhegan Associates and Monhegan Museum of Art and History for collaborating on this year's extensive series of lectures and workshops. We'd also like to thank the Monhegan House for their generous support in the series. The next event in this series will be a presentation by artist and educator Leith McDonald entitled Island Inspiration, which will take place right here next Thursday, 7:30 p.m., July 30th, 7:30 p.m. And a big thank you to all of you for coming out tonight and crowding into this room. At least the temperature isn't as bad as it has been. That can be brutal. It's your support that makes these lectures possible, so thank you. We encourage you to support stewardship of Monhegan's wildlands by becoming a member of Monhegan Associates, and to support stewardship of the island's cultural history by becoming a member of the Monhegan Museum. You can go to the Monhegan Associates website and click on the donate button, and you can do the same thing on the Monhegan Museum website, where we got technology out here. Or if you're up at the museum store, you can fill out a form and become a member on the spot right up there. So before I introduce our speaker, I want to let you know we'll be taking questions after the talk. It says during / after.
What? Oh yeah.
Like
after the talk, right?
After the talk. Sorry, my crib notes are not. Tonight's lecture will be recorded, and if everything goes well with the technology, they'll be posted on the museum and Monhegan Associates websites within about a week or so. Also, before we start, please remember to silence your cell phones. Don't be that person. And now, it is my great pleasure to introduce you to tonight's speaker, biological oceanographer Jess Kusher. As a naturalist, marine researcher, and filmmaker, Jess spent the summer of 2025 developing a short documentary film about Monhegan's tide pools. In addition to past work studying storm petrel physiology, Jess conducted graduate work detecting algae blooms by satellite. She has presented her work to audiences in the U.S., including as a guest scientist at the Smithsonian National Museum of Natural History, and also at international conferences, including Ocean Sciences in Glasgow, Scotland in early 2026. Currently, Jess is researching the movement of prey fish in the Gulf of Maine and serving as a visiting documentary filmmaker with the Marine Mammal Commission. Jess has been coming out to Monhegan in the summer since childhood. It was the island's rocky intertidal and community that inspired her to pursue marine science research. Tonight, Jess will be speaking to us about Refuge on the Edge of the Sea, Monhegan Island tide pools. So please welcome Jess.
Thank you so much, Kristen, and thank you, Laura and Jen and the incredible team who put this together, and all of you. It is honestly such an honor to look out into the audience and see this crowd. It means the world to me. So let's get into it, shall we? There we go. All right, so I want to start with a disclaimer. Exploring the rocky intertidal poses significant risks to you and the environment. So, please be responsible and practice leave no trace, which is to say, plan ahead and prepare, travel on durable surfaces, not our coral and algae friends. We'll talk about all of this later. And be aware that the ocean is a powerful thing and can be deadly. So a little bit more about me and my connection to Monhegan. I wanted to hopefully inspire some of the younger scientists in the crowd tonight, because one of my most impactful memories as a child on Monhegan was hearing an ecology lecture in this very room by a researcher working on her PhD on Appledore Island, studying gulls. So you can see in the top left corner, this is a group of hooligans. I'm one of them, circa 2007, out on Pebble Beach and exploring the Rocky Intertidal. From there, I did a high school science fair, which is a great way to get your foot in the door if you're trying to learn about science. In college, I studied storm petrels and other seabirds, spent a whole summer up in the Bay of Fundy. It was even colder and less technology than Monhegan. And then in graduate school, I did a lot of different projects. So this is, helping with horseshoe crab surveys. We've got me also doing some additional intertidal surveying. My final theses were in both biology and film. This is a documentary film in component that I filmed on an Ohio winter, river. It was very, very cold. In graduate school, I presented my work locally to non-scientific and scientific audiences. This talk will be primarily for the non-scientific audience, but for those of you who have questions, come at me, let's go. Recently, I've helped dissect dolphins, so cetacean intestines for microplastics. I presented my work at the Smithsonian. I presented in Glasgow, we just talked about that. And you can't quite see some of the images down here, but that's me with my head in a tide pool taking some of the photos you'll see today. So one of our goals for today's talk is to think like a marine biologist. This is going to be an interactive talk. As every good student knows, the more you engage with the presenter, the better you do at the end of the day and you get a good grade. So I'm not saying that someone's going to come out with a gold star, but you could. So here we've got an image of the Rocky Intertidal looking down at a crack. And as a marine biologist, my head goes to, let's identify the species, right? We have in the bottom corner here a blue mussel. We have a common name, mermaid's hair. We have periwinkle, Littorina, which is actually an invasive species. They've been here since the 1800s, but not native. And we have what could be small sessile invertebrates. So that's a tiny organism without a spine, so a hard shell, that once it settles onto a rock, it is stuck there for its entire life. And that's one of the things that makes the intertidal so interesting, because you have organisms that can move, like the snail, but you also have a whole slew of organisms that once they have settled, they're trapped. And how are they going to adapt in their environment and withstand incredible pressure? So what can't we see from here? There's probably a temperature gradient because we're going down into a crack. So if you've stuck your hand into a tide pool, kind of at the bottom underneath maybe a rock, it'll be a little bit colder than at the top. I've monitored out here with my temperature sensing thermometer, different temperatures ranging from like 2 degrees to 5 degrees in difference at the bottom of a tide pool to the top of a tide pool. And we're looking at, tide pools that range from a foot deep to, maybe 3 feet deep there. And it all has to do with that shading. So put your hat on. You're now a marine biologist for this talk. So here, some of you might recognize this location. It's Squeaker Cove. We've got a diagram just getting us all on the same page about where the Rocky Intertidal starts and where it ends. We have the lower intertidal, which is where at low tide, the water meets the shore. We have a mid-tide range in the middle here. And then we also have a high tide mark, also a little bit higher up, as well as a splash zone. So here at Squeaker Cove, we have some interesting input. We have fresh water. There's actually a small creek that runs here in the earlier season on Monhegan. So back in May, we had fresh water running into this crack. We also have the input of fresh water like rain from above, and that creates what we call as scientists a brackish water environment. So you have the meaning of salty water from the ocean and fresh water from onshore. We also have nutrients that run off from the woods. What I hope you take away from this in part is that the Rocky Intertidal, while it is technically, these 20 meters, 50 meters or so of zone, it's really interconnected with the rest of the island and the ocean on the whole. So looking over to the other side of my screen, you have the sense that this is a 3-dimensional space. So going closer towards the ocean, we're running up a crack here. That's actually me for scale. You have some of this lower water line. So that's technically the lower intertidal. We have some of this upper space. Now, organisms have to make the choice, if we can say that they get to make that choice of where they're going to settle. So we have some like barnacles, which are sessile, right? And once the high tide comes in, a big spring tide with the highest water we see all summer, and that little larvae settles on a rock, it is stuck there. And unfortunately, if that means it's above the point where any water will reach it on any other tide, that barnacle is toast. It's not going to survive. But the lucky ones will settle where they're going to reach water twice a day at high tide, right? The thing about the upper intertidal is that real estate is plentiful. There's a lot of empty rock space because no one wants to live there, so to say, right? You have increased visibility though from seagulls, from, maybe somebody harvesting mussels, whatever it may be, you're at a higher risk for that. You're also at a higher risk for desiccation stress. So that's what we were just talking about with the barnacle that is never going to see water. It's going to be trapped up there in the heat of the day, and eventually it will die, in part from heat stress and in part from hypoxia, which, just like in humans, is a lack of oxygen, right? All these organisms in the intertidal, for the most part, some of them have lungs, but a lot of them are dependent on doing gas exchange through their gills or other membranes. And so without that water, bringing them food, bringing them cool water from the ocean, bringing them nutrients, they also don't have any oxygen. And that's a terrible way to die. I know this is going to be a grim talk. You're talking to a marine biologist. The bright news is that there are areas, like I've circled this little crack right here. That's a really nice one, or up underneath this rock, sort of under a ledge, where you have more of that lower intertidal environment, right? Real estate is a premium there, and competition is fierce. You've got mussels growing on top of other mussels, right? You've got barnacles growing on top of mussels. You've got very little exposed rock if everyone's at their happiest, most developed out space. It is important that we note that here on Monhegan and in the Gulf of Maine at large, we're losing a lot of the predators. So like sea stars, like some of these other grazers, maybe urchins, that would normally help clear out some of the real estate and allow us to have a balance of species. So when you look at a tide pool on Monhegan or here in the Rocky Intertidal, you might see it dominated by one species. And part of that is they got there first and there's nobody to kick them out. So like I mentioned, cracks are really important. Remember, we're like getting on the same page, thinking like a marine biologist, because in a minute, you're all going to help me run the talk for the rest of it. Okay. You're a marine biologist now. So we're in a crack. This is a great place to hide if you're a motile organism, right? Like a fish that can swim down into a deeper area where the water is going to stay there during low tide. This is a great place to hide if you're a crab. You can move up into a rock that is maybe moist enough. You can still do gas exchange on your gills, even though the water is gone. But like I mentioned, we see that there is like a really hot competition for space down here. We've got a more heterogeneous environment, lots of different organisms. It's not one species, versus up here, we might have one carpeting algae because, there's like less competition and they're hanging out up there. Down here in the crack, we have those juvenile fish, like I mentioned. We also have juvenile larvae and other things like mussels that are going to be, more submerged in that subtidal space. Notably, the Gulf of Maine has a lot of commercially important species, right? We have lobster, we have some crab, we have lots of juvenile fish that we would love to catch and eat. And tide pools and the intertidal space around our islands that are getting more nutrients are an area where some of these species spend their time in a nursery space environment, right? So they're potentially more safe hiding under rockweed in a tide pool than they are getting battered around in the waves and getting exposed to potentially more predation risk from seals and bigger fish. So that's something to think about. Now we're going to show a few little clips, and I'd like you guys to think about what you're observing in this clip in particular. So you're right there in the pounding surf, right? And one of the things that I noticed immediately is that the bubbles and the oxygen in the water is intense. It is so turbid by the amount of gas that's getting churned in this cold water, getting knocked into the rock, that you almost can't see through it. Does anybody else have an observation? Brave soul? Yeah.
Might be hard to hold on.
Yeah, that's a really good point, Becca. I didn't plant that, I swear. Yeah, you've got an incredible amount of stress because your little hold fast, right? So the bottom of the algae, these macroalgae or seaweed, as they're commonly known, is like wedged onto the rock. But if there's a really big storm, you might get ripped off, right? And that's when you go to the beach at high tide and you see all kinds of algae ripped up in a big mat and no one wants to swim at Swim Beach anymore. But it's also an area where these organisms that have been waiting for the tide to come back in finally get inundated with all this amazing, highly oxygenated water, right? You've been holding your breath, so to say, or barely able to respirate with whatever little water you're able to retain on your gills, and now you are flooded. Here's another little clip. It's okay. You guys will warm up to me, I'm sure. So in this particular frame, we've got a huge abundance of and a variety of biodiversity of species, right? We've got some sponge hiding down here in this crack. We've got all kinds of rockweed, some of this big brown algae up here on the rocks. If you're starting to look down in the crack, we can see some of this coralline algae down here. And I'll bet if we kept looking, we could find some hydrozoans, some little epiphytes. We might be able to see ever so faintly a little larval guy or isopod swim by. Something that I want to point over here, can anybody see this little light purple color?
Yeah.
Yeah, so remember that because that's a really cool piece of algae and we're going to talk about that more in a minute. So we've talked a little bit about how hard it is to make a living, as biologists say, in this space, right? You just have to effectively survive, and then you're waiting on the ocean to bring you food, oxygen, water, all the things you need to make it through. Here we have an image of some blue mussels, which are really amazing when it comes to thinking about adaptations. As all of you know, bivalves, right? They have two shells here that come together in a really tight seal. They're actually retaining some water inside of there so that they can continue to do respiration at low tide when they're not exposed to awesome ocean water. And they hold it really, really tight to prevent desiccation stress, but also potentially to prevent predation, right? If you're that mussel that's kind of hanging open, you're the first one to go. We also have down here these really super strong threads. They're called byssal threads. And these are compared to silk or spiderweb silk as some of the strongest organic fibers on Earth because of the way they're able to withstand the pushing and pulling, churning of the waves. And as you can see, these mussels have recruited, as we say in biology, they've recruited onto this rock substrate, but they've also recruited onto each other. In a number of species of bivalves, like oysters, they will affix onto a rock or something else that has high carbon calcium in its shell. So like a friend oyster or another mussel. So another thing that you can't see inside of the mussel at low tide and all the time is are these really specialized proteins, which are actually found in most eukaryotes, right? If not all eukaryotes. So heat shock proteins, this one's heat shock protein 90, are these super tiny little molecules that work to prevent the protein in your body from degrading when it comes under heat stress. So these mussels are sitting out in the hot sun on a terrible day, and just like how you would want to cook them and put them in a pot and kill them, they can withstand some really significantly warm temperatures. And this specific transport protein holds them all together and keeps them from desiccating and totally stressing out and dying. The other thing that's like a more interesting recent piece of literature is that I mentioned there's some water hiding in these shells and they're holding it tight up against them. They're continuing to respirate while they're in the low tide. So they're breathing in oxygen and breathing out carbon dioxide. So what's actually happening inside of the shell is it's becoming more and more acidic. And by show of hands, who's heard of ocean acidification? Yeah, it's something we think about, especially with coral reefs, because of the breakdown of the calcium carbonate on the reef. But these guys are also made out of calcium carbonate. And what's happening in a way is sort of like when you take a Tums after a really like powerful dairy-filled meal and you need to balance everything out in your stomach. They're actually doing a bit of pH buffer inside of their shells by breaking down their own shell. Whether or not they have a choice is, to be decided, but they're breaking down their own shell as a means to reduce some of this acidity and make it more basic in there. So they're a happier mussel on the whole. An area of interesting future study that I believe is already in the works is the impact of having some of these shellfish beds on the surrounding area, right? So maybe ocean acidification can be buffered in a way by, starting more aquaculture with these guys and others. Something I have to bring up because we've been talking a whole lot about heat is the recent heat in the Gulf of Maine. Show of hands, who felt it recently? Yeah, it was really, really hot, and that was above land. In the water, it's all, more mellowed out because of the specific heat of water. But marine heat waves are these movements, you could think of them as like a temperature pattern, where the water is on average 3 to 4 degrees above normal in Celsius for at least 5 days. And so this is something that's very well documented here in the Gulf of Maine, but also all over the world as a significant stressor in some of these environments because organisms like our fish, like our nudibranchs, which we'll talk about in a minute, they're super well timed to what we call a photo period. So just like how we're all used to the way things are, and then we hit daylight savings time and we're thrown off whack, just how birds know because the day is shorter or the day is longer, they can time their flights, their migrations. Organisms in the ocean also time their lives to when the sun is, when we have a longer day versus a shorter day. And so they might be planning on spawning, so like releasing their eggs and sperm when they know that the conditions are going to be just right for their sensitive little eggs to survive what is otherwise a really hostile environment, serious waves, all sorts of organisms that want to eat their larvae, like whales, like other fish. And then a heat wave happens. And they're the sensitive little organism that is barely making it on a good day, right? You're broadcast spawning millions of eggs, hoping that a few of them survive. So heat waves are really bad for our fisheries, but also for us, for the people who care about the ocean. And it's important to keep an eye on them. This is a temperature forecast from the National Oceanic and Atmospheric Administration, so where I worked last year. And you can see this is a chart that shows different months of the year. So we have, this is sort of the warmer months, July through September, October, November, December, and then we go into January, February, March, April, May, June. And this is looking ahead. So this is this fall, this like end of summer, later into the winter, fall. And we can see here the chart is showing the top of South America and then all the way up the eastern seaboard. Maine is up here at the top. And if we're looking over here at this forecast, which is based off of a lot of predictors, including a NOAA buoy that's only a few miles, by the crow flies off the coast of Monhegan, you can see that, on average, we're seeing some temperature anomaly. So actually kind of colder in some regions than we would expect to see off the Gulf of Maine, kind of going this way towards the Labrador. Based on this average over, 1994 to 2023. And what does that actually mean? Because we look a little closer to the Gulf of Maine and it is this brighter orange color. So we are seeing a higher sea surface temperature. It just means that as oceanographers, we're really interested in understanding better how these changing ocean temperatures are going to impact our fisheries, going to impact the way that we live and interact with the ocean. And, especially larval dispersal, because if you want to harvest a lobster in 10 years, you have to care about that initial, life stage. Over here, we actually have another chart, and this is looking at marine heat waves, MHW, from the end of June. And so you can see these dots actually represent the onset of different marine heat wave events. we have a number of marine heat waves events off the coast of Maine here in red that began in June. So that could be a very short-term event, right? It could have been, a few days at the end of June. It could have been the entire month of June. But these are just things as oceanographers that we care a lot about. And we care about more than just the Gulf of Maine and more than just a tide pool on Monhegan, because the ocean is super interconnected, right? The Gulf Stream moving up from Florida, coming all the way up to the top of the northeastern area over here, it's all interconnected. And so whatever's happening in one part of the ocean can influence everywhere else. Now that we're really thinking about being oceanographers and being marine biologists, I want to tell you my primary aims. I know, we're so far into the talk and now we're talking about primary aims. So the goal of this project was to document the current species and biodiversity and the conditions on Monhegan, and to archive different videos for future comparison. As we're seeing the conditions change, it's important to understand where they are now, because we don't have a ton to compare to looking back. And I also want to develop a short communication conveying the different findings of my work. Here's your promise, nudibranch, after listening to me talk. So nudibranchs are sea slugs, as they're often called. And these were found grazing on a spring tide back in June. I had seen 3 nudibranchs my entire life coming out to Monhegan, and then I found over 20 in a day. So it might be one of those spawning events that I was talking about a moment ago. Let's watch that again. So also in this video, you can see some of this red algae down here. And kind of in the background, I pointed out to you the purple, the light purple in a slide previously. This is Chondrus, which is a type of red algae, and it fluoresces a bright purple. I know it looks like reflection in this photo, but there are more photos in the rest of this presentation. And that is actually indicative of it releasing its gametes. So just like how the nudibranchs were all appearing at the spring tide out of nowhere, the algae is on that similar photo period cycle of releasing at the same time. Here we have a really beautiful video going through some green algae in an upper tidal zone tide pool. So this one actually might have a decent amount of brackish water in it. Our green algae do well in brackish and fresh water. And if you think about this area, and the idea that every other breath you take is coming from the ocean, from phytoplankton and algae in the ocean that are doing photosynthesis just like trees, really makes you appreciate how productive and important these tiny ecosystems are in the larger scheme of things. Okay, marine biologists, now let's talk about the 3 groups of macroalgae. So you've heard of algae and you think of it as the slime on the underside of your boat. Yeah, that's algae, but algae is also, these larger structures, macroalgae. So we have 3 different main groups. We have red, brown, and green. The reds are the Chondrus I just described. It's dulse. It's all the beautiful red carpets you see when you look out at the rocks at Lobster Cove. The browns are our rockweed, or our other, all the different fucuses. We have kelp, so sugar kelp. And then our greens are a lot of different ulva species. They're called sea lettuce for a reason. They look a lot like lettuce and you can actually eat them. Okay, so marine biologists, what do you notice about this picture? Any takers? You've warmed up now.
Very colorful.
Yeah, it is very colorful. So those pigments, just like chlorophyll, right? We think about chlorophyll in the trees as they change color in the fall. There are other pigments that also participate in photosynthesis, and we're seeing those represented here in these bright reds and these beautiful brown greens. Here's some things you may have noticed. We have down here some coralline red algae that's kind of encrusting on this rock. It's a really beautiful feathery fan. We have a periwinkle. We also have a mussel hiding up here in the top of the frame. What I'm trying to help you hopefully realize and think about is that while you might be focused on the most beautiful red algae center frame in a tide pool, there's 1 million things happening at once. And the more you stop and think and look, the more you start to realize what all is going on. So here's more of that beautiful purple, it's technically a red algae that I was just talking about, and more mussels scattered along it, just so you have the frame of reference. Okay, as promised, here's the Chondrus with the iridescent, beautiful purple that we see on the tips here. You can also see all kinds of little isopods floating by, swimming. Can be very chaotic in a tide pool. So as described, tide pools are an important shelter space, nursery space for all sorts of larvae. Here we have an image of a lobster larvae, and here we have some sea urchin larvae. Of course, in the Gulf of Maine, we have green urchins. They're making a comeback and might be making a reappearance in the aquaculture trade. Who notices the crab?
Oh, yeah.
Yeah, so in this picture or in this video, we can see a whole lot of fuzz in the water, right? We also notice that crab. That fuzz could be all sorts of different larvae. We'd have to look at under a microscope to understand what it could be. It could be isopods, a whole different variety. So I mentioned lobster, and I would be like in trouble with every lobster biologist ever if I talked about tide pools as the end all be all in the lobster life cycle. I see Jess in the back. She has actually the background in lobster science. So here we have a juvenile lobster, unfortunately dead, that I found at Swim Beach earlier this year. Off of Swim Beach and in the Monhegan Harbor, we actually have a pretty significant patch of eelgrass, which is a premium location for this sort of like earlier life stage of lobsters. They really enjoy boundary spaces. So where we go from, you know, a pebbly bottom to grass, or from pebbly to solid rock. And who's to say precisely why, but eelgrass is an area of incredible interest for, you know, lobstermen and for conservationists because of the way it shelters these early species, but also people who want to study carbon sequestration in the ocean, right? Eelgrass is actually a grass, it's not an algae, although it creates a lot of habitat space for different algae that grow on it, and unfortunately also slime mold. Yeah, I'll bet you guys didn't know there are fungi in the ocean. There are a lot of fungi in the ocean, and there's so many marine bacteria, we have not even classified all of them yet. So here we have a slime mold that's making a really big appearance in some of our eelgrass beds. And if you've looked at some of the strands that get washed up on the beach on Monhegan, they do have this characteristic brown patchiness, which causes them to fragment and break and have just really sad die-off. But there are all kinds of great isopods that feed on some of the epiphytes that grow on this algae. We have all sorts of snails that grow on this, that eat everything that grows on this algae. It is truly just an amazing space to swim through if you're ever feeling brave enough to stick your head in the water. It is, however, just like our shell fisheries, getting impacted by some of these heat waves, right? I'm sure some of you folks from Florida especially have heard about harmful algal blooms. So this is a map that is updated regularly about where the shellfish is getting closed in these different shell fisheries around Monhegan. This is the current map from earlier today. We have some green, so those are safe to eat the shellfish, and we have some yellow, which is and then orange and red. And some of these models, if they're not going out and actually sampling the water to identify the harmful algal blooms, are based off of water temperature, known nutrient loading, and some of our satellite products, which is what I did my master's work on. So I'm happy to talk to you about that later. But so that's just to say, going back to this idea of marine heat waves and their impact on the overall ecosystem, it's impacting the food you want to eat. I know some of you like oysters in this room. So here we have another video looking down into a tide pool, and this is at low tide. We can see some movement from this periwinkle on the top right. You see a lot of this fuzzy stuff that you're all thinking, God, this girl loves fuzzy stuff. It's so boring. And you can see a mussel. We can see all sorts of little barnacles. We see the whole spectrum of algae, right? We've got green, we've got red, we've got this beautiful encrusting algae here. We've got some brown. It's really just a total rainbow of a tide pool. However, some of that crusty stuff, some of that fuzzy stuff is invasive, and we're going to get into that right now. So these are a whole series of slides on the invasive algae of Maine, which are becoming more prevalent from some of our marine heat waves, as a lot of them are from more tropical climates. And I've only included algae that I've personally seen in and around Monhegan. So we have dead man's fingers, which I'm sure you guys are familiar with. It's a delightful sponge to play with and throw at your friends. Here we have seed potatoes, which you might see more of in a moment in another video. Keep your eyes open. We have different types of rockweed. Specifically, this is the serrated rockweed. We have this siphoned featherweed, which all of these, I should add, are from the Pacific for the most part, a lot of them around Japan. We believe that they've come to the United States by way of ballast water. We also think that's how periwinkles got here. We've got this beautiful red seaweed, but you can imagine if you're trying to pull up a line and it's covered in algae, that's a real pain. We have this bryozoan, which is a colonial organism, just like how we think about coral as not just one polyp, it's a whole organism, or we think about a beehive as not just one bee, but a whole organism. Bryozoans and these tunicates that I'm about to show you are these colonial organisms where they are filter feeding and covering some of our algae. Specifically, this lacy encrusting bryozoan is covering our kelps and preventing them from being able to photosynthesize appropriately. If you're familiar with the story of kelp on the West Coast, you can have a trophic cascade where, for example, on the West Coast, if you don't have sea otters eating the urchins, then the urchins go out of control, they kill off the kelp beds, and this critical habitat space is lost. Here we have the example of a bryozoan killing off kelp in the Gulf of Maine, and who knows, they could be lost. For now, they're okay. They're actually making a good comeback. Then we have a number of tunicates. This is a carpet tunicate. You can see it kind of looks like snot totally covering a rock. But if you were snorkeling, I promise you might look at it like this and think that could be a sponge. It's not. We have this beautiful jelly crust tunicate, which we primarily see in tide pools on Monhegan in this sort of darker gray form. And it looks a lot like if you like chia seeds and you make chia seed pudding, it looks like you've smeared that on a rock. And it feels pretty similar too. Then we have this orange sheath or chain tunicate, and this is another like absolutely beautiful thing to see. But unfortunately, it is an invasive species and it's taking up that premium real estate from other organisms. And we also have this other really beautiful tunicate, but again, invasive, this golden star tunicate, which you often see in more of this gray form up here. We finally got to the fuzz, guys. These are hydrozoans, which cover often fucus. This is specifically a sea oak hydrozoan, but they're also creating this fuzz on our eelgrass in the harbor and all over our tide pools now, much more than they did 2 years ago when this project began. Keep your eyes peeled for anything that we might have just talked about. Who sees the sea potato? Yes, 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 right there. Sea potatoes, for those of you who can't see the laser pointer in the top left, they look like big orbs on the rock. We also saw some dead man's fingers in this. We can see a whole slew of Litorina are periwinkles, which actually are commercially harvested by some people. And it's a beautiful tide pool on the whole. Here we have another image. This is one of our tunicates covering and just totally smothering a bed of mussels. We have some hydrozoans hanging from the top of this undercut rock here. We also have some growing all over these mussels in a tide pool here. And the truth is that some of these are native species, and it's great to have a diversity of species in a tide pool. It's true that we don't have sea stars the way we used to, so it might be okay for some of our mussels to die off. Right? Then it creates more habitat space for everybody else in the tide pool. But the truth of the matter is, we don't fully understand what's going on here. This is like new and growing at a much higher rate than I would have personally expected. And hydrozoans, although not necessarily these, can contain stinging cells. So you never know what you're going to touch. They could be releasing some kind of toxic compound in the water. You don't know what you're getting your hands into. So it's something that we need to continue to monitor. I don't mean to alarm you guys, but it's something that we need to understand. And that's part of the reason of this study, is to take photographic and video evidence of what's going on now so we can compare it to what's happening next year and the year after that and the year after that, ad nauseam. So here are two images of sea stars. The one in the left is a brittle star fragment that I actually found in the gravel on the wharf. My running theory is that it came off of one of the lobster traps earlier this year that was sitting out there. And here's one down deep subtidal in a secret tide pool that maybe someday I'll show you, my marine biologist friends. And so the thing about sea stars in the Gulf of Maine is that maybe you've heard about sea star wasting disease on the West Coast. We're aware that that could be happening here in the Gulf of Maine, and that it is exacerbated by some of these stronger heat wave events, because with a warmer water, right, the metabolism of these organisms increases, and so they're respirating more, they're expending more energy, they are more stressed out, so to say, and it makes them more susceptible to some of these parasites, diseases that are causing them to die off in really, really high numbers. I know from talking to a lot of you in this room, you can remember a time where the wharf used to be covered below the waterline in, you know, sea stars and urchins. And it's up to us now to understand what we currently have and look ahead to how it is changing. So maybe we can create some positive change and maybe see them come back, right? Yeah, a northern sea star. I know you guys are going to point that out to me. So as we previously sort of described, we do have green urchins in the Gulf of Maine. These are all urchins that I found on Monhegan. This is my nifty little bin. If you're ever out tide pooling with me, I like to put a critter in the bin so you can see it without any distracting rock or algae. And here we have two smaller specimens that I found earlier this year. My hand is super swollen in this photo because I found that guy back in May, went back to the same pool, could not find them. Decent chance that the water has gotten warmer and they've all moved deeper into the subtidal space. So maybe out of tide pools deeper off the rocks. Could also be that they were predated, because it feels like the gulls, that's the one thing they can catch and eat, right? So urchins were actually fished really dramatically in the Gulf of Maine in the late 80s into the early 2000s. We see this really big peak and then a total drop off of the fishery. I know, again, from talking to some of you in this room, that's a good thing, that's a bad thing, right? It brings in money to the economy. It also might be a bad thing because maybe we overfished them, we weren't regulating them appropriately. We are seeing potentially some come back, and that's also a blessing and a curse. If they're filling a lobster trap, it's making it harder to bring your lobster to market. But if they're able to clear off some of this rock space and create more habitat for all these like array of different species, maybe that creates a better environment for some of our young lobster. So we have to appreciate that there is balance in the system, and hopefully we can regain some of that balance. Now, you guys earned some really peaceful sea anemones. These are the big orange ones. Here's a fun fact for you, because you're all marine biologists and you ought to know, sea anemones can detach from their rock. They're actually motile organisms and they can swim. So if they don't like this crack, wait till it's high tide. You may never see them in that crack ever again. So here are some major takeaways from this talk, right? These ecosystems are interconnected. We have lobster and juvenile larvae, all sorts of great species hanging out in a tide pool as a safe space and then moving out into the ocean where they might be caught and become your dinner. We also have runoff from the land, everything we're doing here on Monhegan that's going to enter these tide pools, these sensitive environments, and impact whatever is going on inside of them. And there's interconnectedness, right, with these marine heat waves and how stressors we might add to the environment could be exacerbated, right? We could see things get worse because the organisms are already stressed. There is an opportunity, as hopefully displayed through my talk, to leverage modern technology like underwater video to document the biodiversity, so the different array of species in these environments, and then to analyze them later as we're able to train potentially AI models to start identifying species and count every single one of the little barnacles, so it's not an undergraduate's job for their entire life. And we also know from what we've discussed today that change is happening, right? We're seeing marine heat waves here in the Gulf of Maine. We are feeling some of the heat waves on land, and we're seeing an increase in non-native species. We could be seeing an increase in disease, and all this, like I said, is getting pushed to a breaking point as we're seeing hotter and hotter temperatures. But it's exciting to study the rocky intertidal, because if there's one group of organisms that knows how to withstand desiccation stress and heat, it is these guys who are dealing with it twice a day, or at least once a day. And the present work represents an ongoing effort to document these local conditions. And I hope you guys will stick along with the rest of this project as it continues. Now I want to end on a quote by Rachel Carson from Under the Sea Wind: "To stand at the edge of the sea, to sense the ebb and flow of the tides, to feel the breath of a mist moving over a great salt marsh, is to have knowledge of things that are as nearly eternal as any earthly life can be." So I want to thank a lot of people. I have to recognize that part of the present work was conducted through support from the National Sea Grant College Program John A. Knaus Fellowship, which is how I spent my last year in Washington, D.C., pushing papers and barely getting to shove my head in a tide pool, just barely. And I want to thank the Monhegan Associates, the Monhegan Museum, and the Monhegan House for their generous support of the lecture series. And I especially want to thank all of you because as somebody who has grown up coming back to this island over and over again and who understands the magic in the way that I hope all of you do, it means so much to come full circle and to stand in this room in front of all of you and to share some of what I've been up to. So thank you. I am so glad to take any questions.
All right, I'm gonna prepare yourselves. I'm gonna turn on one of the lights so that Jess can see.
There are people.
I just have a question about,
Yeah.
you know, it's so important and funding is very important. And how much of funding to study this is from fishing industry versus the government? Or where does the money come from if it's so important?
That is a very interesting question. So part of this work was conducted while I was doing a fellowship in Washington, D.C. for people who've completed their master's, PhD, or JD and are focused on marine science. So the part of this work was not explicitly funded by any funding agency. It was more of, I got explicit permission to work on an ongoing project and come out to Monhegan and document some of this work. In terms of like interest in the Rocky intertidal, I'd say there's a lot more on the West Coast. And the University of Maine does a lot too. You know, I think that there's a lot of focused money on lobster larval development, for example. And in terms of biodiversity, a lot of biodiversity has been done. I think there's less of an interest now in maybe doing more natural history work, which is, I think, where this more closely aligns in terms of thinking about the array, than there is asking very pointed, specific questions. So, you know, we'll see. I'm very hopeful that there will be funding in the future for this project to grow and outlive me. So, thank you.
Very interesting.
Thank you so much.
I see incredible colors in your filming. All of this is genuine filming, you looking, you filming, etc. There's no enhancement, there's no reddening or oranging or greening.
So in the very first picture, at the very beginning of my slideshow, I took that picture and I said, there's something wrong with my camera sensor because this is so vibrant. And then I looked back at where I was and it was one of those cloudy, foggy days where the ambient, like diffused light just made everything pop. And it was one of those moments where I was like, oh, I'm like so taken by this. But no, when you look at this image, this is not edited for color. I've decreased the brightness some just so that you can see like a little bit more of the array, but that would be against my ethics as a documentarian.
It's beautiful.
Thank you, Doris.
Were all those video clips shot off Monhegan?
Yes. Yes, the photos were attributed if they weren't from Monhegan, but everything else was.
What camera did you use?
So I used multiple different cameras. Some of the underwater filming was done with GoPro. Some of it was done with an iPhone in a waterproof case. Some of it was done with my Sony A7S III. So depends. Yes.
So the invasives that you talked about, how much impact do they have on the non-invasives that they're impeding on? And is there anything that can be done to prevent it, more invasives from coming in through the way you're saying they come in?
Yeah, that is a good question. Let me repeat it for the Zoom because I realize I haven't been doing that. Dawn wanted to know how the invasives are impacting our native species and if there's a way to prevent their continued spread. So my answer as a biologist is always, it depends. So I think you'd have to look like maybe on a case-by-case basis, depending on the species, right? Because we have some species like our fucus, our rockweed, that is so pervasive that, you know, coming in with a serrated fucus is probably not changing too much the space it's able to take up in the rocky intertidal. But for some of these more sensitive species, it's definitely having an impact, right? We see these colonial tunicates that are just covering a mussel bed, and you think, wow, if that could take over like even an even larger amount of space, like could we have a problem in the future where, you know, the mussels aren't able to make it, or they're able to make it only in subtidal conditions or in upper tidal conditions where that tunicate can't survive, right? Can they find a different ecological niche, whether that's like colder water or less water, where they're able to survive but the tunicate isn't? And so going to the second part of your question, it's hard to say, right? We know from some research around trophic cascades, like we talked about the sea otters, that the reintroduction of a keystone species like a sea otter can have a significant effect on the environment, right? We look at the West Coast and Morro Bay in particular, and the reintroduction of sea otters and the way that they were able to eat the urchins, and then the kelp beds came back. So, you know, we might someday find funding in the Gulf of Maine to target a specific population and maybe bring back sea stars, for example, or bring back sea urchins through any number of different reintroduction techniques. And maybe through their pressure as predators on the system, the sea star, of course, eating the mussels, and the urchins, of course, grazing some of the algae, maybe we would see a change in the species composition because we have that kind of like bully on the playground, keeping everyone in line. Yeah. Yes.
So, you know, last year's exhibit at the museum used the artistic record as a land-based biological database. Right, and we talked about that. And then we were talking about the changes that most of us, you know, time sort of remember what was in the dock, or you know, how many urchins were there, whatever. Talk about what they remember kids. But is there any, like here or elsewhere, a similar data that wasn't collected to be data that might give you a baseline?
I am so glad you asked that question. It's like you planted it. So for the folks online, this gentleman just asked if there is another set of data that's not technically data, like a scientist in the field with a clipboard, that we could use to better understand the baseline, like historic information of where Monhegan's biodiversity was in the past. And he used the reference of like the museum utilizing past paintings of Monhegan artists as an understanding of what the ecology used to be on land. Great question. So right now I'm actually working with Matt Weber, and we have co-written a grant that potentially could pay lobstermen and people with significant marine ecological knowledge of Monhegan to sit down and talk about how it used to be and their understanding of what it was in terms of like abundance of different species, where they were finding them, et cetera. Ideally, if a separate grant pulls through that I won't go into too much detail on, it could be broader than that, and it could include community members like yourself, which would be awesome, because I do think that the way people care about Monhegan and care about coming back, they have that knowledge and they have that memory, and potentially you guys have a photo album that actually could fill in some data gaps. So.
Great. Well, thanks everybody for all the great questions. And let's thank